Okay. My name is Jorma Jurkinen. I'm a biologist who's been studying carcinogens and mutagens for many years. And I became interested in antibiotics through work of Colorectal and their potential to influence cancer. And this is a paper that I was giving a talk on at the Gynecology and Genetics Conference 2020. And unfortunately the video didn't display, so I'm trying to make this up now. Samir Kalgatki discovered that reactive oxygen was increased when you gave 3 different antibiotics. Lipid peroxidation was increased, and this was quite a long-running effect. And glutathione decreased. Well, both of those, reactive oxygen and lipid peroxide, are involved in aging and cancer and mutations. And so Kalgatki discovered that these doses, quinolones, aminoglycosides, and beta-lactams, cause mitochondrial dysfunction and reactive oxygen overproduction in mammalian cells. And they demonstrated that these can lead to oxidative damage in DNA, proteins, and membrane lipids. If you look at the mitochondrial genome, as a circular genome, as typical of bacteria, there are a lot of diseases that are potentially produced by mutations in these. Cardiomyopathy, prostate cancer, encephalomyopathy, deafness, diabetes, and other diseases. So we don't want to mutate this thing. These are some of the proteins that are produced mutated, and also polypeptides. The number is quite significant. If you do a computer search of the mutations caused that are linked to cancers, there's 3,717 DNA mutations that are linked to cancers. Okay, so antibiotics induce changes to mitochondria that results in potential contributions to carcinogenesis. Antibiotic killers slow the growth of bacteria. The mitochondria is an ancient alpha proteobacteria and has become an endosymbiont in higher life forms with critical functions. And so this has the potential to impact cancer. Mitochondrial DNA defects are a relatively common cause of inherited disease and have been implicated in both aging and cancer. Mitochondrial DNA encodes essential subunits of the mitochondrial respiratory chain, and defects result in impaired oxidative phosphorylation. This, of course, can result in glycolysis, which cancer cells like very much. So cancer is listed as one of the diseases associated with mitochondrial dysfunction. So do antibiotics have these effects? Do they induce mitochondrial malfunction, damage, or rupture, prevent them from faithful reproduction, cause mutations in genetic material damage, interfere with tumor suppressor genes, create microbiome dysbiosis, harm the immune system, increase the Warburg effect, statistically significant associations? So quinolones inhibit bacterial DNA synthesis and result in rapid cell death. They can be expected to do collateral damage to mitochondria and the human microbiome. This group contains afloxacin, norfloxacin, ciprofloxacin, moxifloxacin. The expectation is to obstruct mitochondrial replication. Aminoglycosides create holes in the outer cell walls of bacteria, suggesting mitochondria and microbiome might be at risk of similar damage. Beta-lactams inhibit cell wall synthesis in bacteria, and by inference inhibit cell wall synthesis in mitochondria and microbiomes, thereby damaging lipid membranes and obstructing reproduction. Tetracyclines are of special concern. They're used on cattle and humans. They're chronic residue because we eat meat. Mode of action is by binding to the ribosome, preventing attachment of the aminoacyl transfer RNA. And it simultaneously inhibits other steps of protein biosynthesis, and we can expect this in the mitochondria. There's also a problem for clinicians. Antibiotics have been identified in some cases as causing aneuploidy, and clastogens include other substances as well, X-rays, viruses, and drugs. These decrease cell division rate, break chromosomes, cause chromosome deficiency, deletions, rearrangements, and changing chromosome numbers, chromosome aberrations, and sister chromosome exchanges, and micronuclei. And if you find these, this predicts cancer. The lipid peroxide induces cancer mutations. So what are the other sources of reactive oxygen that are at issue here? Well, the exogenous ones are foods, drugs, pollutants, xenobiotics, and toxins. And so these can be expected to compound any that are produced by antibiotics. Reactive oxygen species in cancer, or elevated rates of reactive oxygen species, have been detected in almost all cancers, where they promote many aspects of tumor development and progression. Reactive oxygen impacts various diseases. DNA reactive oxygen forms reactive oxygen adducts, and a single and double strand breaks, and DNA damage triggers P53 increase. However, the P53 is subject to mutation, and this is linked to colorectal cancers especially. These mutations in particular, KRAS, APC, and BRAF, and P53. You can determine the reactive oxygen by a stress test by assay of 8-oxo-DG. Genetic alterations in colorectal cancer, these are very similar to the ones that reactive oxygen is capable of producing. TP or P53 mutations are prevalent in sporadic cancers. The proportion of tumors with somatic P53 mutations is indicated. You can see that a great many cancers have TP53 mutations. So mutation of the P53 gene is very critical. Where the loci of these are is also important. And for each cancer type, there seems to be a particular locus that is affected. Methylation status of CpG dinucleotides in the substituted segment of the human P53 knock-in, puppy and mouse syndrome. Methylated cytosines with CpG dinucleotides along the DNA binding domain of TP53, tumor suppressor gene, are the single most significant mutational target in human cancers. These are the pathways to colorectal cancer. ROS can induce a number of these mutations. Fusobacterium nucleatum is normally an oral bacteria that lives mostly in harmony in our body, but it can go become active in inducing inflammation and suppressing host immunity in the gut. And it can lead to effects very similar to any antibiotic effect. But other bacteria and microbes can do the opposite. They can reduce inflammatory factors. For example, bifidobacteria. So F. nucleatum can induce immune suppression of gut mucosa by suppressing function of immune cells, such as macrophages, T cells, and natural killer cells. So the lesson is don't mess with healthy microbiota. Cardiology and mitochondria. Mitochondria are incredibly numerous in heart tissue, and so any dysfunction in them or anything affecting their function is likely to lead to heart disease. These are diseases that have a link to mitochondria: atherosclerosis, ischemia, reperfusion, IR injury, hypertension, diabetes, cardiac hypertrophy, and heart failure. And the UBC study has found that there's 2.4 times increase in mitral valve regurgitation with antibiotic exposure. These are due potentially to uncontrolled production of ROS, reactive oxygen. SOS 2018 found that mitochondria form large networks which regulate metabolism. Optimal function is achieved through balance between mitochondrial fusion and mitochondrial fission. However, that function depends on quality control by the process of mitophagy. And I think that that's an area that we need to get very knowledgeable in. Okay, sources of lipid peroxide. There's mitochondria is listed, activated macrophages, and neutrophils, and NADPH. The reactive oxygen species then go on to produce lipid peroxidation. Other causes of lipid peroxide increase: taking analgesics like aspirin, NSAIDs, naproxen, indomethacin, diclofenac. Being male hypertensive smokers, oophorectomized in pregnant women, eclampsia and preeclampsia, and ochratoxin and mycotoxin in cereal. So you know your causes, and then you'll have a better idea of what's going on. Red meat and processed meat, you can see here in the right-hand side, it has lipid peroxidation, and it forms mutagens, which go on to make adducts in cancer in the DNA. The ethanol DNA adducts in particular are derived from the 4-hydroxy-2-nonanol, and this is found in other cancers as well, at least one that I know of. And in alcohol carcinogenesis, reactive oxygen lipid peroxidation, inflammation, and immune suppression are all involved. And you can see the chain on the right-hand side here as well. Reactive oxidation and lipid peroxidation are very much involved in the DNA adduct. Lipid peroxide is it's mutagenic metabolite trans-4-hydroxy-2-nonanol. I found this in several cancers. It forms DNA adducts at P53 249 codon locus. This is associated with hepatocellular carcinoma. So this is a smoking gun that antibiotics are potentially linked to cancer. So hepatocellular carcinoma has been identified at that locus. That locus is also associated with human papillomavirus through Aspergillus bread mold mutagen. It may mutate other tumor suppressor genes as well, and also we need to look at other loci, as I mentioned before. Lipid peroxide, tumor suppressor gene VHL mutation is linked to hereditary kidney cell carcinoma or renal cell carcinoma, and possibly LP-ROS is induced but not proven. Lipid peroxide ROS induced. Okay, we haven't proven that connection. P53 overexpression is linked to RCC, renal cell carcinoma, possibly linked to lipid peroxide. Hydroxy-2-nonanol, as I already mentioned, and in ethanol-linked cancers. Aerobic glycolysis and DNA damage are consistent with promoting carcinogenesis. This is known as the Warburg effect. When the oxidative phosphorylation is diminished, as it would be with damaged mitochondria, the fallback metabolism is aerobic glycolysis or anaerobic glycolysis, and cancer cells favor that very much. So what's the evidence from epidemiology? Well, Kilkinen has discovered relative risk of 1.31 to 1.48 for prostate, increases for breast, lung is higher, and endocrine has quite a range. So those cancers are increased. Relative risk for exposures greater than 1.5 for 6 treatments of antibiotics for skin, duodenum, pancreas, kidney, bladder, male genitals, thyroid, myeloma, and leukemia. Newman says there's an increased risk of bowel cancer, and Belisar found prolonged use leads to fatal breast cancer. Ben Morrissey found use of penicillin associated with elevated risk of esophageal, gastric, and pancreatic cancers. And he adjusted those for age. He found gastric cancers increased 1.2 to 1.8. Lung cancers increased 1.3 to 1.6. Prostate cancer was modestly increased with quinolones, sulfonamides, and tetracyclines. Breast cancer modestly associated with exposure to sulfonamides, but there was no association between use of antivirals and antifungals. The next thing I want to talk about is the immune system and the antibiotics' effect on the immune system. Well, short-chain fatty acids are derived from bacterial fermentation of dietary fibers in the colonic lumen. The SCFAs, butyrate, propionate, and acetate promote intestinal epithelial barrier function and regulate the host mucosal immune system. Well, if you knock those out, you lose your efficacy of your immune system. So, antibiotics, TKO, the short-chain fatty acid bacteria. Without them, the immune system goes down. Immune depression has many medical consequences, opens the door to adventitious infections, pathogenic viruses, bacteria, worms, and some carcinogenic. Viruses and antibiotic-resistant bacterial infections risk. And the next 6 slides, aside from a little slideshow that I put on, enlist organisms that may cause cancer that can be facilitated by depressed immune systems. And if they do this, it will lead to increased population cancer incidence. But first, I want to talk a little bit about things that make your immune system less effective. PFAs are potent immunotoxins, meaning they impair immune responses in both humans and animals. These are fluorocarbons. It's been found that they reduce vaccine efficiency and antibody response in humans at concentrations rarely found in our blood. So this is an immune knockdown. So if you're polluted with these things, then these antibiotic effects could be worse because your immune system isn't functioning properly. If you're also polluted with other pesticides and other chemicals, you may be more liable to immune knockdown as well. And this study by Crocker shows that very nicely. So here are the cancers that you can get from viruses that take advantage of opportunistic immune knockdown. And bacteria, viruses, and also worms, schistosomes and liver flukes, Clonorchis, Opisthorchis, and Hematobium. Okay, so inflammation is a big factor in cancer. So these infections can lead to chronic inflammation. And this will cause cancer cells to proliferate, and it will also order stem cancer cells from the stem repository in the bone. Reactive oxygen, ROS, can induce the formation of oxidative DNA lesion products, but we've already said that, so carry on. So prevention of infection-associated cancers, Paolo Bonanni came up with this strategy. Chemical hepatocarcinogens, avoidance of exposure, chemoprevention, diet, drugs, health education, regulation, vaccine, and general prophylaxis. Bacterial infection can also lead to cancer. Chronic inflammation will lead to production of bacterial metabolites that are carcinogenic. Bile salts may cause colonic cell proliferation, and rutin may be metabolized into mutagens by resident colonic flora. Bacteroides can produce fecapentines, and hydroxyl radicals attack DNA. Colobactin causes DNA breaks, and BTF genotoxins increase interleukin-17 inflammatory cytokines, etc.